Good morning, good evening, good afternoon, everyone. Welcome to iConnect Talks, Connecting the World and the Universe. My name is Professor Martin Thuo, coming to you from the eastern side of the United States at North Carolina State University. This month, we have been exploring the theme of bioengineering, and last time we heard from Peter Wang from University of Southern California. Today, we are very fortunate to have Johan Foster from University of British Columbia. And next week, we'll have Professor Wang from Queensland, and then Professor Kerich from University of Cape Town. For today's talk, I'll be assisted by two panelists, Zhu Li from Fudan and Jiaxuan Zhang from University of Electronic Science and Technology in China. And our ex-challenger is going to be Ji Wan from Peking University. A little bit about Professor Johan Foster. Professor Johan Foster and I have known each other for many years, starting out when he was working with liquid crystals, mesogenic liquid crystals with Vance William at Simon Fraser University. After that, he took a detour out of North American continent into Europe, where he worked and delved into the world of polymers and biopolymers specifically, both in the Netherlands and in Switzerland. After that, where he started his career actually as a faculty in Switzerland. Then after that, he came back to the US and was a professor for quite some time at University of Virginia Tech University, from where he recently moved back home to Vancouver to the University of British Columbia. And his main focus has been on nanocellulose and cellulose-based materials and derivatives of such materials. He'll be sharing part of his work in that area with us today. And it's my pleasure to introduce a friend, a great scientist, and a very well-established member in the cellulose world, Professor Johan Foster. The floor is yours.
Great, thank you, Martin. I will share my slides. Yes, thank you very much, and welcome everybody from all around the world. Good morning, good evening, good afternoon. Today, as Martin said, I will be talking to you from the beautiful British Columbia here on the west coast of Canada. I will tell you a few stories about how we use rod-shaped fillers to modify a variety of different polymers, and we're going to probably even get to some titanium metals by the end of the talk, so a wide range. Before I really start to talk, I need to acknowledge my collaborators throughout the years, and really the students that did all of this work, right? I mean, as Martin can attest to, and all the other professors that we work with, we get up and get to talk about this, but really it is the students that we've had in our group, the visitors, the collaborators that we've had that have allowed this to happen, and combine that with the funding that we've had throughout the years to really enable this sort of research. Okay, so here in British Columbia and many places around the world, we like to utilize things from the forest. And I use that very broadly because we use cellulose, we use lignin, we use hemicellulose for a wide variety of materials. Now we can think about what we can do with these materials, all the way from energy, so we can burn it, for example. That's a very kind of a low cost or low return on your investment. And then all the way up to biomaterials or implantable materials or electronics that we can make out of these. And it's important to have a wide variety of these types of applications, right? Because you need to utilize these materials in a variety of ways. So to talk today, we are going to focus on the use of cellulose nanocrystals, so this rod-shaped filler that I'll describe, for a few applications. In particular, biomaterials. Now, in this case, biomaterials can mean two different things, right? It can be biosourced, which it actually is, but in this case, I mean an application inside the body, okay? So biomedical. And then we're going to get into using these for a kind of a different application in lightweighting and changing materials properties of both polymers, and then we're going to get to titanium. So cellulose nanomaterials. Derived from nature, so you can get them from things like wood. Okay, so a tree is made up of cellulose and hemicellulose and lignin. You can also get this cellulose from cotton, or from even other creatures, from bacteria, from tunicates from the ocean. But regardless of that source, here we have the example of a tree. If you break that tree down into its constituent pieces, and then degrade those components, you can end up with a couple different types of nanomaterials. On the left, we show cellulose nanocrystals. We've also called them cellulose nanowhiskers, or nanocrystalline cellulose in the past. So the community has really tried to focus on using cellulose nanocrystals as the term we use. Rod-shaped, from say 100 to a few thousand nanometers long, and say 5 to 20 nanometers wide. So really, think about the shape of a pencil, but in nanoform. There's a wide variety of other sources you can potentially use from this. If you degrade this enzymatically, you could end up more in kind of spaghetti-like material, but we are going to focus on this rod, very stiff-like material. Okay. So again, we can talk about these being rod-shaped. You can draw them as kind of rice-shaped even. You could draw them as pencil-shaped, all very stiff and isotropically shaped materials. So long, thin materials that we can use. Okay. So wide variety of sources, wide variety of chemistries on the surface. So the cellulose nanocrystal, as the name suggests, is made out of cellulose. So that repeat unit that you see in the bottom right, it's a lot of hydroxyl groups. We can put a few charges on the surface depending on how we make that material. So sulfate groups, for example, if you used sulfuric acid to dissolve away the rest of the tree, you could put phosphate groups on the surface, which I'll talk about a little bit later. You can hydrolyze that the primary alcohol to a carboxylic acid. You can, and then use all sorts of chemistry. So my background is in chemistry, and so modifying those hydroxyl groups is always very fun, because that allows us to do all sorts of tricks with these. Okay, but what I want to get kind of tell you is that we can modify the surface. We can keep that shape, and that will become very important when we want to use these as a useful reinforcing tool within polymers and other materials. Okay. One thing that we should note is that the thermal stability of these is quite important. Now, for some applications, like implants in the body, well, hopefully your body will never get above about 37, maybe 40 degrees Celsius. But for other applications, say reinforcing a polymer or reinforcing something that might get very hot, the thermal stability of cellulose can be very important. So we undertook this is a paper we published maybe about 5 years ago now, where we looked at the surface charges on this nanocellulose, and if we modified those surface charges, we can change the dispersibility, so the ability of this to be dispersed into a solvent, and with that comes a change in thermal stability. Okay. So 3 cases here in the top left. H means that there's only a hydrogen, those hydroxyl groups are just hydrogens. The P means they're phosphate groups, and the S means they're sulfate groups. So you can see that the H, the solid line, is the most thermally stable. The phosphate and the sulfates are less stable, but with that comes some sort of dispersibility. So you need to be able to balance that, because one key part about having this fantastic cellulose nanorods is their ability to be put into other materials. Okay. So we can compare these to other things that we would like to put into, say, a polymer. When you talk about a tire on your car, for example, reinforcing that rubber with, say, Kevlar or carbon, fantastic materials. In the case of, say, steel wire, really strong, fantastic material, but is very, very heavy. So on a car, that's not so important. But if you were trying to reinforce materials in, say, an aircraft or something that flies, you want to be very cognizant of how much that weighs. So at the bottom, we've got cellulose nanocrystals, which have a density of about 1.4, 1.5, have a fantastic Young's modulus, anywhere between, say, 100 and several hundred gigapascals, a high surface area, and potentially a low cost. So these can be a nice material. They're being produced all around the world now. So by the ton in several plants. These are in Scandinavia, some in Canada and the United States, and there's a few in Asia right now that are producing cellulose nanocrystals anywhere from, well, kilograms to tons. Okay, so what do we want to do with these? My group has been working on cellulose nanocrystals, as Professor Stowe introduced, for years, and so we've had a lot of very cool applications. We've worked in cortical electrodes, so this is implants in the brain, which we'll talk about. We'll talk about biological implants, but you can think about using that rod-shaped filler to imbibe interesting properties on other materials. So functionalizing the outside to make them transparent, but also conductive. So you can make a replacement for indium tin oxide on phones. You could reinforce tires, like I mentioned previously. And so we'll talk about a few of these applications. The first one, mechanically responsive. So I often say that my group works on smart materials. Now, in this case, I put smart in quotation marks. All I mean by smart is that they are responsive. So you take the material and you do something to them. You give it a stimuli. So that could be adding temperature, shining light on it, implanting it in the body. You do something that changes it. And that change comes in a prescribed manner. So that could be a change in color or stiffness, or release of a drug, for example. And you can do lots of things with this. You can coat materials like you see on the left. You can release those on command. You can make shape memory polymers. You can make things that change color. Now, in the bottom right, I show the Terminator from Terminator 2. We're not quite to that level yet, maybe in a few years, but we really like to think we can make some very interesting materials that respond to stimuli. Okay. We do this because of academic curiosity. It's very interesting. There's huge potential for making new materials, for making innovative materials. We can really add value to very simple polymers by increasing their mechanical properties. And hopefully we want to make a better quality of life in the case of the biomedical devices. I will talk about a few materials later that are not smart. I jokingly call these dumb materials. But it's very difficult to get funding for dumb materials. So we say smart approaches to not so smart materials. All I mean by that is that not all of my materials will change in a prescribed manner when you apply a stimuli. So the inspiration for this stimuli responsive materials came from nature. On the left here, we have a sea cucumber. Now, these are little creatures that look like a small cucumber that crawls along the bottom of the ocean. They are edible. You can eat the inside of the little sea cucumber. But the sea cucumber has a very interesting defense mechanism. When it's scared, the whole sea cucumber goes very, very stiff. Okay, and so about 15 years ago now, or 20 years ago, a couple of biologists figured out what was going on inside of the sea cucumber's skin. It's mostly soft material, collagen, fibrillin, a lot of water, of course, but it was reinforced by small fibers. In the relaxed state, so when it wasn't scared, those fibers are not really interacting. When it is scared, it excretes a protein called tensilin into its skin, which cross-links the fibers within its skin, creates a network of those fibers, and the whole animal becomes stiff. This happens very quickly within the sea cucumber. Now, when we were trying to take the pictures of these sea cucumbers, we actually had to rent a sea cucumber and try this. I can attest that it happens very quickly. It becomes stiff very quickly, but it takes a long time for the little animal to relax. Okay, and I'm not sure if that's because we didn't play nice music for it, or make it give it a relaxed environment. But this doesn't necessarily happen very quickly in both directions in nature. So we want to do this in the lab. What we do is we take a soft modulus polymer and we reinforce it with our cellulose nanocrystals. Now, if we can get those cellulose nanocrystals to interact, we can make a stiff material. And if we can turn off those interactions, decouple those cellulose nanocrystals, we can make something that's very soft. So exactly that structure that we mimic of the sea cucumber. So in the bottom left, we have a logarithmic scale of stiffness versus volume fraction of the cellulose nanocrystals we put into that polymer. Okay, in the top line, the blue line, those cellulose nanocrystals are interacting, so they're stuck together. In the simplest case, this is held together by hydrogen bonds. Those hydroxyl groups on the surface of the cellulose nanocrystals are interacting. Okay. Now, if we decouple those interactions, it becomes much softer. So that's down to the red case. Okay. So that means they're not interacting. They're still a stiff, and they're inside of a soft polymer. So they're still reinforcing a little bit, but not nearly as much as when they're stuck together. Okay. So now we can create a material that, when it is dry, those cellulose nanocrystals are stuck together, and when it's wet, it becomes much, much softer. So order of magnitude change in the stiffness of this material. And it is very reversible. Again, not very quickly. It is controlled by the diffusion of water into the system, and then the drying of the water out of the system to couple and decouple those interactions. Okay. For the first application we want to talk about with these switching materials, I'll introduce the area of the brain-computer interface. Now, this has been talked about for years and years. Elon Musk has a company that is looking into interfacing computers in the brain. That's Neuralink. But when we first started this research, in the bottom left, we have a lady. This is not research from my lab. This is previous research. But this lady has a cortical electrode on the top of her head, and she can move that robot arm and feed herself. Okay? Still quite rudimentary, but this is taking an array of electrodes, implanting it into the brain, and allowing the brain to interact with the computer. Okay, so really recording firing of neurons within the brain on a single neuron level. Absolutely fantastic research. You can think about, yes, interacting with a computer. You could talk about interacting with your cell phone. That's probably not what you would want to do. This is major surgery. So think about treating things like Parkinson's disease, for example, other neurological diseases. These electrodes were made of things like tungsten and steel, fantastic materials. But the problem was that they were failing. So again, major surgery, but the electrodes would fail after just a few weeks. When they took the electrodes out, they saw that there was scar tissue around the electrode. Now, that's tough because your electrode fails. You don't want to reimplant a new one. And the hypothesis was it was the difference in stiffness between your electrode very, very stiff material like tungsten and steel inside of your brain, which is very, very soft. Think the texture of a jello or a gelatin. And that mechanical mismatch was a problem. So hopefully you see where we're going with this. What we want to do is take a very stiff material. It needs to be very stiff because you need to poke it into the brain. It needs to penetrate the brain tissue. So as something that is as stiff as, say, a credit card. And then after you implant it, you don't need it to be stiff. So you want it much, much softer. Okay. So we want that mechanical switching. So we pattern an electrode on the surface of our switchable material. We implant it into the brain, and we see how long we can make this interact with brain tissue. Okay. You might ask, how do we actually make the material like this? Well, it's actually pretty straightforward. Once you figure out the ins and outs of this, you take a solution of the polymer. So this particular case that we're showing here is polyvinyl alcohol. So dissolve that in water, disperse cellulose nanocrystals within that, combine them, make sure that they don't flock, or they don't fall out of solution, or they don't start to aggregate. Cast it, evaporate the solvent, and you're left with a film. Okay. Now, depending on the size of the cellulose nanocrystal and the amount of the cellulose nanocrystal we put into this material, we can tune the stiffness in the stiff state and in the soft state. Okay. So depending on the type of brain tissue, or if we wanted to go into other types of tissue, we can tune the mechanical properties of those to be able to exist in different situations. Okay. You might ask, well, are the cellulose nanocrystals actually dispersed? It looks like they are, yes. It's very difficult to image the cellulose nanocrystals within a polymer matrix because, well, cellulose is just a biological polymer, right? I mean, it's carbon, hydrogen, and oxygen, and a polymer like polyvinyl alcohol is also just carbon, hydrogen, and oxygen. So it's you don't get very good contrast within these materials. And so images like this, where we show mostly dispersed cellulose nanocrystals within this material, seem to indicate that they're very well dispersed. We can't see objects that are larger than the diffraction limit of light, so we know there's not huge aggregates within this material. And the proof is in the fact that we get this fantastic reinforcement of the polymer. Okay. So because it was early morning here in Vancouver, I decided not to put any implant pictures in. We work with a group in the United States where they do implants of these materials into brains. This is a 16-week endpoint, so 4 months later. And I'll walk you through these results. So this is thinly sliced sections of brain that is then stained for certain types of cells. So neuron cells, that's what you want to see. That's your brain working. And 3 different cases. We have silicone. Okay, very stiff material, doesn't change its mechanical properties. On the right, we have our adaptive nanocomposite. That changes its mechanical properties quite dramatically. And in the middle, we have a polymer-coated wire. So same polymer as our adaptive nanocomposite, but it's just coating a stiff metal wire. And that was just to answer the question whether it was simply the surface chemistry of our adaptive nanocomposite and not the switching. So at least I can see it on my screen. You can see a void space around the neuron in the silicone-coated wire, and the silicone and the polymer-coated wire, and you can see growth right up to the surface in our adaptive nanocomposite. That's great. In the paper, we've quantified how close this is and how many cells there are. Astrocyte growth is what you don't want to see. That's essentially the defense mechanisms at work. And so in the case of the silicone and the polymer-coated wire, you see significant astrocyte growth. In the adaptive nanocomposite, you do see astrocyte growth. It's not a perfect material. And in fact, while you're poking something into the tissue, you're causing inflammation at the site of implantation. And you will always do that with any sort of implantable material. So want to be aware. Yes, this is fantastic. We feel like we've really solved part of that problem of a material that can adapt to brain tissue and be better, but it's not perfect. Okay. So what we want to be able to do is deal with that inflammation. So we're going to combine that idea of a mechanically switchable material and now start to work in other aspects onto those cellulose nanocrystals, onto that rod-shaped filler, to be able to deal with other problems, in this case with implantation. So we made a first generation of this material very simply by mixing in anti-inflammatory drugs into our adaptive nanocomposite. So combining those drugs with the CNCs, they adhere to the CNCs, or they don't chemically bond to the CNCs in this case, although we do have systems where we do that now. Okay, so two different drugs. On the top we have curcumin, and on the bottom it's resveratrol. Curcumin would be in turmeric, so again a biomaterial, and resveratrol would be an anti-inflammatory that's in red wine. Okay. So we can show that we've got release over time. That's fantastic. We can show that really we wanted a burst release within the first 48 hours because we wanted the site of inflammation to be to have those anti-inflammatories most dramatically. In subsequent generations of these materials, we've been able to tune the release to have burst kinetics, so simplified by diffusion, and then other mechanisms where we attach the drugs to the surface of the cellulose nanocrystal and can release those over the course of anywhere between 72 hours and up to 3 weeks. Okay, so with that, kind of showed you one story where we're able to tackle a mechanical problem within the body, create a material with our rod-shaped fillers that dealt with a problem with those materials. Okay, so implants in the brain. Next story. This is with a former PhD student, Sandra Camarero, who's a professor now in Spain, where we cured the knee. Okay, so inside of your knee, the bones in your leg are held apart by cartilage. It's a fantastic material, and lots of people have problems with it, myself included. Okay, so my mom's side of the family has horrible knees. They've had to have knee cartilage replacement, and so I felt like here's a problem that would be relevant to my family and many families around the world. So I was able to find the data for the United States. So that's in 2007 to 2009. Almost a quarter of adults were diagnosed with some form of arthritis. So something to do with your joints. Okay, so this is a huge problem, and what can we do to fix it? So cartilage itself is very particular. Now, the one problem is that it's structured in a very specific way. So here is a diagram that we made of what cartilage in the knee would look like. On the top, you have the superficial zone, which is characterized by kind of fibers that run parallel to the bone surface, and also that kind of dictates that the cells that grow in that region are also somewhat aligned with that, parallel with that surface. In the middle, you have kind of unaligned fibers, unaligned cells, and an area that's rich in glycoaminoglycans, or GAGs. That really helps retain water within this structure. And then in the bottom, in that deep zone, it's characterized by fibers that are aligned perpendicular to that bone surface and has a lot of hydroxyapatite. So that allows it to interface with that bone. Okay, so just by itself, natural cartilage is very particular. The other problem is that not all cartilage is created equal. So my cartilage would be different than, say, Dr. Thwo's. He grew up in a different area. Maybe he drank milk as a kid, or maybe he did more running, or all of these sorts of different things as you grow up, or your genetics will dictate how thick your cartilage is and how stiff your cartilage is. So if you damage that cartilage, the problem is that that damaged area doesn't support the surrounding tissue, and that damage propagates. The other problem is that if you implant a material that is not like the surrounding tissue, it can abrade the surrounding tissue, or it cannot support the surrounding tissue. So you have to tune this to an individual. Okay, so what do we want to do? Well, we want to utilize our cellulose nanocrystals to be able to tune those mechanical properties, to be able to tune the chemistry of these of this system to make something that we can tune to an individual. So a 3-layer system where we mimic the cartilage that's in nature. A superficial layer, a middle layer, and a deep layer, again with that same sort of alignment. And we imbibe this with the chemistry by tuning the chemistry on the surface of the cellulose nanocrystals. So now we're utilizing 2 different aspects of these cellulose nanocrystals. That reinforcing aspect, so using that rod-shaped filler, but then also that chemistry on the surface to be able to make it mimic the surrounding tissue. This is all held together with polylactic acid for very specific reasons, which we'll get to. So lots going on here. This was a lot of work by Sandra for her PhD. But essentially what we did was we took 3 different layers. So I'll walk you through this. In the top left, we have 3 different layers. We were able to make unaligned foams and aligned foams. So mimicking that deep layer, the middle layer, and the superficial layer. Now, aligning in one direction versus the other, this was using thermally induced phase separation to give an elongated structure. It also allowed us to, depending on the interactions between the polymer, the solvent, and the cellulose nanocrystals, we were able to have the cellulose nanocrystals present at the surface of the material or embedded within the walls of the material. This allows tuning of the mechanical properties, but also if we want to express, or if we want to show the surrounding tissue what that surface chemistry is, we can do it. We took those layers, we fused them together. Now this was non-trivial because cartilage for these implants is approximately 1 centimeter thick. So you need to get 3 layers of foam within that thin foam. Sorry, 3 different layers within that 1 layer of foam. We used solvent welding to get these to stick together, and then we pre-grew hydroxyapatite onto these. So we were the first group that actually created cellulose nanocrystals with phosphates on them. And that was simply to be able to get those calcium phosphate pre-growth of hydroxyapatite in that bottom layer so we could integrate it with bone. On a completely side note, it turns out that phosphated cellulose nanocrystals were actually very advantageous in their thermal properties. So I kind of told you about that a little bit later. But originally we had created them simply to have phosphate groups on the surface that we could pre-grow hydroxyapatite. Okay, the bottom right, we show that we can grow hydroxyapatite in the deep zone, but not in the other layers, and that is the presence of the phosphate groups on the cellulose nanocrystals. The sulfate groups or just hydroxyl groups on the surface do not cause hydroxyapatite growth. Okay. So one very interesting thing, obviously I've kind of been alluding to this, we can tune the mechanical properties. We can change the thickness of each one of those layers. And we can make multi-layer scaffolds. Outside of the body, we can actually pre-grow your cartilage. And I suppose it was very satisfying, is that over the course of a few weeks, the mechanical properties, because we did this in the right way, because we gave it the right mechanical environment, The mechanical properties of the cartilage after 2 weeks or 4 weeks mimicked the natural tissue very nicely. That's very good. Of course, we can show how that tissue grows. Just in the interest of time, I'll kind of just show this and say that we can pre-grow cartilage. The cells differentiate exactly the way we want them to be able to have that grow in a bioreactor outside the body for very advantageous implant into the body. Okay, so there is an example of being able to take a rod-shaped filler, use it in a biomaterial. Now, you shouldn't use cellulose nanocrystals in every implantable material, but in this case, it really gave us a handle to be able to make cartilage that was tuned to a person's natural cartilage. Okay, and that was very important. Sorry. Now we're in the process of bringing this to approval, working with a company to be able to do this commercially. So, interestingly, we're doing trials in pigs. So, if you ever wonder what sort of animal has knees just like humans, turns out that it's pigs. And so we are doing implants of our cartilage mimics into pigs. Okay, so we've implanted into the body. We've put it into the brain. That's very, very soft material. We can make a sponge-like material, can implant it into the knee. But outside of the body, what can we do with this? Okay, so I used to jokingly say that with all of the tools we have in our lab, we can take cellulose nanocrystals and put them into almost any polymer. Okay. Using these tricks within the lab, we can take a dispersion of the cellulose nanocrystals. We can mix them with polymers. We can make a film or make a foam. And you could do this in a variety of ways. But easiest inside of the lab is mixing those solvents together, evaporating it. The problem with that is that making it any bigger, so scaling it up industrially, is not very viable. Using all that solvent is slow and very costly. So what can we do? So we have a couple of very interesting methods to utilize those cellulose nanocrystals to reinforce materials, but get them into polymers that are very difficult to, say, cast. So in this case, I asked a couple of my industrial friends, and they said, well, if you can get cellulose nanocrystals into polyethylene or polypropylene, that would be best, because those two polymers make up the vast majority of polymers used worldwide by volume. Okay, so in the case here, we have an ethylene molecule that we put through a catalyst, and we end up with a wide variety of materials. So a lot of the bags you use, the pipes that are in the cities, the chemical bottles you use in the lab are probably made of polyethylene. If you do this in very, very interesting ways, you can end up with ultra high molecular weight polyethylene. So this is really long polyethylene. We're talking about 1 million molecular weights. You can end up in materials that could be replacements for body armor. Okay, so this is Dyneema if it was made by DSM, and if it was made by Honeywell, it would be called Spectra, but really high-end polymers. So a huge, like a wide variety of things you can do with polyethylene. But can we do it different or better with cellulose nanocrystals? So years ago, we tried to do this with our solvent methods. We took a cellulose nanocrystal dispersion. We made an aceto gel. So we replaced the water with acetone. So that's a nice gel-like material on the left. We imbibed in our polyethylene. So we dissolved our polyethylene and put that inside and pressed it. Fantastic results, which I'll show here. Neat LDP has a Young's modulus of about 400 MPa. With just 7% of our cellulose nanocrystals, we can get up to 1600 MPa, so 1.6 gigapascals. So you can really take a low end, so low density polyethylene, and reinforce it with these cellulose nanocrystals. So huge mechanical reinforcement using this. Now, like I said, using this solvent, this is a multi-step way to get to reinforced polyethylene, not industrially viable. But it shows the possibility that if you do this in the right way, you can get to a reinforced polyethylene. Okay? So a very low end polymer that you do these tricks with and make it much, much better, kind of into the engineering polymer regime. Okay, but we wanted to do it better. So we took our cellulose nanocrystals and we put a catalyst and a co-catalyst on the surface, and then just pumped in polyethylene, or sorry, pumped in ethylene into the solution to make a CNC polyethylene composite. So here's something that we could do very industrially. You take your cellulose nanocrystals, okay? You functionalize with a polymer that's already used industrially. Now, granted, this metallocene is not generally used at scale, but this was what we could do kind of easily in the lab. So we functionalize the surface of the cellulose nanocrystal with our metallocene. We make sure it's covalently bound, so we wash away any other solvent. And now with the presence of a co-catalyst, we can make some very interesting material. So here we have AFM. Hopefully want to show that we've got cellulose nanocrystals in there. Again, very difficult to show. We know they're in there because we know they didn't go anywhere else. But our cellulose nanocrystals, so this is AFM, atomic force microscopy, showing our cellulose nanocrystals on the left. The comparison to that very, very high-end, ultra-high molecular weight polyethylene in the center and our composite. To be honest, you can't really see the presence of the cellulose nanocrystals. We know it's different than the ultra-high molecular weight polyethylene, but to actually, again, see the presence of those is very difficult. In this case, keep in mind, we've taken those cellulose nanocrystals, we modified them with the catalyst, and then when we polymerize it, the polymer completely coats the cellulose nanocrystals. So that's good in the case of the composite, but very difficult in actually the imaging. Okay. So, as a lot of people would say, the proof is in the pudding. The proof is in whether you can get mechanical reinforcement. So, two different cases here. We have ultra high molecular weight polyethylene, sort of the best polyethylene that's out there, and we have high density polyethylene, which is also a very widely used polyethylene. And we compare that to our cellulose nanocrystal reinforced polyethylene. So we have storage modulus as a function of temperature. So this is dynamic mechanical analysis. And we can show that at most usable temperatures, we have a much stiffer material. Not much stiffer. We have slightly more stiff material, which shows the possibility of taking, again, a very cheap material. Neither of these polyethylenes are cheap, but other ones are, and create a reinforced composite that allows you to use cheap materials to make higher end product. Okay, so this really shows the possibility of scale. So remember, right at the beginning of this talk, I said, well, we want to be able to utilize these materials in a wide variety of applications. Really high end, we have our biocomposites. We have our implantable materials that are very expensive, but you won't use very much of them. Not that many people need implants in the brain. There are quite a few people that need cartilage replacement, but again, that is a very small amount. But when we can talk about reinforcing polyethylene or polypropylene, we're talking about millions of tons, millions of kilotons per year, right? So megatons. So huge potential scale and application for these. Great. One of my students, Priya, she said, well, polyethylene was good, but we really need to be able to, do this in a wide variety of manners. She wanted to reinforce plastics for the aircraft industry or with for the automobile industry. Okay. She said, well, okay, we can put catalysts on there, but can we put them through an extruder? And this is problematic. Okay, so if you just take a container full of cellulose nanocrystals and a container full of your polymer, you dump them into extruder. Oh, it's messy. Whoever owns the extruder would probably yell at you a lot because it would create a big mess. Okay, long time cleaning the extruder. We tried it a couple times. Showed that it really didn't work. You can mix pulps and cellulose fibers with these polymers, but they require some sort of modification. Okay, you can modify the surface with silanes. You can modify them with salts. You can graft polymers on them. You can use mixing aids to allow that extrusion. Lots of great papers out there from anywhere between 10 and say 30 years ago. Okay, the problem with using these aids is they disrupt the interactions between your fibers. That's why it allows this to work. It allows it to be dispersed. But I told you in the first half of this talk that all of that great reinforcement we get is because of those interactions. How do we get our cellulose nanocrystals or our fibers into a polymer without disrupting that? Well, we need to do it in kind of interesting ways, because we don't, bad things happen. So in the case of our LDPE, if you take our low density polyethylene and you take some cellulose nanocrystals and you put them through the extruder, in the best case, it looks like the image in the top right. In the worst case, the whole thing turns black and it's a big mess. But in the best case, you can see sort of that degraded cellulose within that polymer matrix. The cellulose is aggregating, it's burning. This is problematic. So, two things: sticking together, so the aggregation, and the thermal stability of our cellulose becomes problematic. With melt extrusion, by its very nature, you need to melt that material to get it to mix. So in the case of polyethylene, you're probably talking about 160, 180 degrees on the low end for mixing this material. For other polymers, like the polyamides, you need to be at 250, 270 degrees Celsius to be able to mix these. So the thermal stability of your polymer is okay, but the thermal stability of your cellulose, your filler, is hugely problematic. Okay, so ours starts to turn brown or turn black, and you don't get the reinforcement. So we want to solve this. In this case, we're going to use PA11, so polyamide 11, structure in the center there. We've tried a few other ones now. This was our first one, simply because it was bio-based, semi-crystalline. We could potentially recycle it and biodegrade it, and it was commonly used for replacements in the automotive industry. So the problem was we were worried about the thermal stability of our cellulose within our polymer. So we thought, well, maybe we can utilize the thermal stability of the polymer to stop the degradation of the cellulose nanocrystal. So our first attempt was mixing in polymers into a milk compounder on the right. Do it in the wrong ways, the cellulose nanocrystals start to yellow and brown. If you premix this, so if you put them into a mill, you bang them, the cellulose nanocrystals and the polymer all together, the polymer starts to coat the cellulose nanocrystals. Then when you put it into a hot press or into an extruder, it seems to be that the cellulose nanocrystals are buffered from degradation by the presence of the polymer around it. Okay, so if you premix it, essentially what you're doing is you're using the polymer as a coating. Well, it's also the coating and the matrix, which allows the fairly quick dispersion within the larger matrix. Okay, so if you direct mix them, they start to aggregate. Cellulose loves to stick together. as that the thermal stability isn't really buffered, you get degradation. Okay. So it's a very interesting trick to be able to take these cellulose nanocrystals and mechanically push them into the polymer at low temperatures, and then use that as a kind of a master batching, as a method to be able to get this into larger polymers. Okay. What happens? Well, we get fantastic mechanical reinforcement of the polymer itself. Again, if you do this in the right way, or if you use the right cellulose nanocrystals, you can get the thermal stability. Not all cellulose nanocrystals are the same. I could give a whole talk on the differences between cellulose nanocrystals. So we used 3 commercially available cellulose nanocrystals. We just give say they're A, B, and C. We can talk about which ones those are later. But in this case, the ones that were C, a lot more charges on their surface, a lot less thermal stability, and they turn black and don't reinforce very well. Okay, but this shows the effect of both ball milling and then compounding of this material to get a reinforced composite. So this is polyamide 11, and drastically better properties than the neat polymer. So again, evidence that that those cellulose nanocrystals are held together and really reinforced. Okay, and we attribute that dispersibility somewhat to the charges on the surface. And if you do this in the wrong way, if you get very, very high, highly charged surfaces, they become less thermally stable. Okay, so with that, I will kind of wrap up this the use of cellulose nanocrystals and polymers, and I'll say that it requires the surface chemistry to be done right. You need to be able to interact with the cellulose, interact with each other, interact with the surrounding polymers, or interact with the human body. And you need to be able to mix that in the right way. If you can't get your cellulose nanocrystals throughout that polymer, then that becomes problematic. Okay, so stability, dispersion, and shape of your nanofiller is very important. If you can control that, if you can get that dispersed into your polymer, you can get a smart material. You can get a reinforced material. So I set up all that, and I will change gears for one last story. All started from a conversation that I had after giving a talk. It's very similar to that. I was at a conference, an American Chemical Society conference, huge conference, and I was talking in the polymer division. And I think after the talk, over drinks, we were saying, well, I can get cellulose nanocrystals into anything. And one of my colleagues that works in the United States mumbled something to me, and he said, "Well, I'll if you can get it into this, we'll that'd be fantastic." And I didn't really hear him, but I said, "Oh yes, I can do that. Of course, I can get it into anything." I meant any polymer. And so the next day, he comes back to me and said, "Well, I'm really excited about you being able to do this. How do we get it into titanium?" And I said, "Oh, did I agree to that? That's well, that was surprising. I probably shouldn't have done that." So this whole project came about by a kind of a misunderstanding each other to be able to reinforce titanium. So titanium is a fantastic material. It's usually formed in nature in its oxide form. So titanium oxides of a variety of sorts. Very high specific strength, corrosion resistance. It is biocompatible, although we'll not talk about that here. It is kind of one of the standards for very, very lightweight materials when we're talking about putting things into space or very high-end aircrafts. Okay. Applications in marine, you can make them very, very nice colors. If you look at very high-end cars, a lot of the exhaust systems would be done out of titanium. Again, very, very high-end applications. And we do this because, titanium by itself is actually is a very nice material. The titanium alloys can be great for bikes. I know my bike has a few titanium alloys on it. But then for very, very high-end applications, you want to get into titanium metal matrix composites. So this is reinforced titanium, and you can get titanium blends. So for example, the gold standard for things that will go, say to Mars on a space shuttle would be Ti-64. So that's a titanium, aluminum, vanadium composite that is kind of have the highest strength, the highest stiffness titanium. Okay. The problem is that making these sort of composites is very, very expensive and often very toxic. So, alloying, the poor wear behavior, and the problems with kind of processing led my colleague to say, "Hey, can we reinforce this with cellulose nanocrystals? Because apparently I claimed that we can reinforce everything." Now, I had a very, very kind of basic idea of using the tricks that we've talked about in this hour about creating a cellulose nanocrystal matrix and putting in titanium around it, which was a horrible, horrible idea. It was never going to work, I suppose. But that's actually what I originally had envisioned. So I actually talked to some people in the field and said, "Hey, here's my wonderful cellulose nanocrystals. How do we use these to reinforce metals?" And they said, "Well, there's some obvious problems here. The stiffness of your material is good, but not compared to metals. And the other problem is the thermal stability is off by an order of magnitude. So our cellulose nanocrystals start to degrade about 150, 180 degrees. And any of these titanium alloys, we're going to start to sinter at 1,000 degrees or 1,200 degrees. So problematic. But all of those tricks we used previously, what can we do with milling these materials together, utilizing cellulose not just as its rod-shaped filler, but as a way to start making a metal matrix composite? Okay. So in this case, we wanted to mix these together. We were wondering whether the cellulose nanocrystals would coat the titanium. And now this is after many, many tricks and many different things we could do. We took attrition milling. So that is mixing the titanium powder and the cellulose nanocrystal powder together, compressing that. So using a cold isostatic press to compress that into a green body. So this would still contain cellulose, maybe not as individual rod-shaped objects. But then the key part here is kind of in the bottom, heating this to 1,300 degrees Celsius for an hour. So me as an organic chemist, as a polymer chemist, when we talk about high-end, high temperature materials, think something like polyether ether ketone, fantastic polymer that is stable up to 400 degrees Celsius. That would be the higher, kind of the higher end of polymers. Cellulose doesn't last that long, and we're heating this to 1,300 degrees. So what does our cellulose nanocrystal, that rod-shaped filler, now that it's mixed into a green body with our titanium, do as we start to do this? Okay, so if you do this in the wrong way, it will act as a source of oxygen, I suppose. But what we found was that the CNCs degrade if you do this in a controlled manner and nucleate the growth of titanium oxide within the titanium. Okay. So if you do this in a controlled way, the oxygen diffuses to these points. Okay. So it's no longer a cellulose nanocrystal. We've degraded it, but it acts as a nucleation point for the growth of titanium oxide within that titanium. they coarsen, so they get bigger and bigger as you do this more and more. That pulls oxygen out of the interstitial sites of our titanium. And now you've got a titanium oxide, which is a ceramic, reinforced titanium composite. Okay, so I mean, I'm telling you what we ended up with. We had to vary the amount of milling time. Okay, so this is prior to the sintering, the milling time of our cellulose nanocrystals with our titanium. Vary the amount of cellulose nanocrystals in this. So you might ask, oh, is it something special about the cellulose nanocrystals, or would it be any polymer that we use to do this, or any cellulose? We have tried other celluloses. So we've tried pulp fibers on kind of the big end. We've tried dissolved cellulose. Doesn't seem to do this same trick. Okay, so it could be the. I personally, I don't know exactly what the reason is, but it could be the crystallinity, could be the size that it coats the cellulose nanocrystals in very nice ways. But we get these very interesting structures. So once you get into the kind of a little bit more cellulose nanocrystal and a little bit more milling time, you start to see the growth, this is after sintering, of these titanium oxide platelets within the titanium matrix. Okay. Good. So the hardness increases as a function of the growth of those titanium oxide platelets within the titanium. I tell you now that they're titanium oxide platelets. That took actually quite a bit of microscopy and quite a bit of testing to figure out that they actually were. So the great thing about this, I told you earlier that imaging our composites was very difficult with cellulose nanocrystals within polymers. Imaging titanium oxide within titanium is fantastic, just because of the contrast between these. Okay, so we were able to digest these materials, look at the, well, in this case, titanium oxygen ratio within that and the rest of the matrix, figure out that that is titanium oxide. Do compression testing. This is actually should be a video on the right, which is very not dramatic and then turns very dramatic in a second. Hopefully. Maybe not. Maybe this is not actually a video. In this case, it really should be. Nope. Of course, it's not playing. All I show is the compressive strength of this these titanium oxides, or sorry, titanium matrix metal matrix composites are actually very, very stiff. And I suppose I'll skip up to kind of what this actually does. When you take attrition milling, you do this to titanium itself or with a few other additives. The oxygen dissolves into the interstitial sites of the titanium, and you get very hard, very brittle materials. When you do it with titanium in CNCs, the CNCs act as those points to basically increase strength and create that metal matrix composite. Okay. So I will conclude and wrap everything up. Really what we want to do in this case is compare to the best out there, and that would be Ti-6-4. We have just as good, if not better, compressive strength in a lot of cases, and in the case of Vickers hardness, so that's the measure in metals, that's the measure of hardness. We have better hardness than the industrial standard. We think this is a fantastic material and really allows us to get to a wide variety of sort of applications. We really think that this was a case of, this is very serendipitous in that this did not reinforce like in any of the other ones. It really is creating a very, very different material, but utilizing those cellulose nanocrystals in a very unique way. So we're doing many more things with this to try to make it better. Mill it with oxygen scavengers, for example. Try to reduce the amount of energy. Maybe do this in a vacuum to be able to tune the properties of this. Okay. So with that, I'll wrap up. And I'll wrap up the same way I started. I'm really thanking the people that did all of these projects. Implantable in the brain was mostly Mehdi Jorfi. In the knee was Sandra Camarero. The composites with catalysts was Keith Hendren. The PA11 was Priya Venkatraman. And then Johnny and Manuel were the ones that did the titanium. Obviously, lots of other people, lots of other collaborators. So I'd like to thank them for all that work, the people that funded this, to the whole ICANX team for the invite, Martin for the introduction, and everyone for listening. Thank you very much.
Thank you very much, Johan, for such a wonderful and long journey into nanocellulose and how we can use them. It's very surprising that cellulose is so ubiquitous and we don't think about how much we can use it. So in continuation, we will first jump into the audience questions, and then we will get back into the panel. So we have two questions. As you would expect, there will be a question from IOR. So we have two questions. The first one is from Fully, from Jian. And the comment is, thanks for your wonderful talk. I'm wondering whether the adaptive nanocellulose can be biodegradable and how long it can take, can it work in vivo? Basically, what is the lifetime? We looked into that.
No, we didn't look at the lifetime. In general, the electrodes in the brain are removed after a set amount of time. That particular composite was polyvinyl alcohol. Our polyvinyl alcohol in that case was slightly cross-linked, so we heated it to about 150 degrees when we pressed it, and so created a few cross-links so it wouldn't dissolve within the body. You didn't want the polymer to roam around the brain. So we very specifically made that non-biodegradable within the body. You generally do not want polymers to degrade inside the brain, as a general statement. But we could make, we have made versions of that are biodegradable. Well, biodegradable in, if you bury it in the soil, but also biodegradable within the body. So this one was not, and I would say the lifetime inside the body was anywhere between say 4 months and 8 months, but not degraded.
Very good. So the next question comes from Ior. How does the surface modification affect the swelling? Again, the biodegradation property of this nanocellulose, and does it also influence the dynamic modulus?
That's actually a very good question, Paul. Paul from Iowa. The surface modification, I would say, does not directly affect the swelling or the amount of the swelling. The surface modification does affect the transport of water in and out of the system, but the amount of swelling is usually dictated by the polymer, the cross-linking of the polymer, for example. That being said, surface modification does affect the speed of the swelling and the transport of water in and out of the system. So not the amount, but yes, the surface charge. And so we've utilized not an adaptive nanocomposite, but the cellulose nanocrystals within membranes for water separation, for example, or water transport through systems.
Okay. Very good. Very, very interesting talks. And so now we're going to get into our panel. And our first panelist is Zhu Li from Fudan University. Actually, this is a reunion of former VT, Virginia Tech people, because she was in Virginia Tech, where she did a master's, then went to Georgia Tech, and then from Georgia Tech, she was in industry for a few years before jumping across the Pacific to start her current position as an assistant professor at Fudan University.
Very nice.
And she's done a pretty amazing work in bio-inspired material, artificial skin, and embedded, or what I would call polymer composite. Great, amazing work with some very interesting papers coming from her group. Then we have Xiaozhen Zhang from University of Electronic Science and Technology. Dr. Zhang did a bachelor's and MS and PhD from China, starting out at the University of Electronic Science and Technology in China for BSMS, followed by a PhD at Peking University, then jumped all the way to EPFL for a postdoc, and then a slight walk home, first stopping in Tokyo, then back at University of Electronics Science and Technology. And he's done pretty amazing work in microelectronics and other applied materials. Our ex-challenger is G1. G1 is actually a PhD student at Peking University, working on some work on microelectronics and applied composite. He has published quite a few papers, very interesting work, even at an age, is a truly rising star, and we're very delighted to have you here. And so we're going to all join into the panel, and we will give our ex-challenger the first chance to ask our speaker questions.
Okay, thanks, Professor Martin, and thanks, Professor Foster, for your wonderful talk. And I have two questions for you. And the first one is, in the neural electrode work, what material do you use to make the polymer conductive at the same time, have great impedance match with the brain tissue, and have no toxicity?
So I kind of glazed over that. The adaptive composite itself is not electrically conductive. We take that adaptive composite, we pattern an electrode on the surface, so a thin gold electrode.
Okay.
And that is the part that's actually electrically conductive in that generation. We have made electrically conductive composites, but they were using silver nanoparticles, and so they were conductive. But the people that wanted to implant these in the brain said, no, you cannot put silver nanoparticles inside the brain. It will. They didn't say it would be toxic, but they said it would definitely not be a good thing inside the brain. So yeah, we used a gold surface, a very thin gold electrode that didn't affect the mechanical properties.
Yeah, okay, thank you. And the second question is that I noticed the main processing method for the composite of cellulose nanocrystal and polymer is drying and then molding. I wonder whether the cellulose nanocrystals can be processed by other methods like laser cutting, spin coating, physical or chemical etching, so that it can be prepared for more shapes and maybe it can have more application scenarios.
So laser sintering is an interesting one. If you do that in the right way, you get just cutting of the polymer, and we have used laser cutting to cut out our composites. But if you use like laser sintering as a writing technique, you can actually get carbonization of the cellulose nanocrystals, which can be very interesting if done in the right way. But in terms of other techniques to get different shapes, we have 3D printed these adaptive materials and the composite materials. Very interestingly, the cellulose nanocrystals, because they're rod shaped, shear thin. So when you put them under shear, they become less viscous. So this actually works quite well for things like 3D printing or extrusion-based materials or methods, right? So we are looking at other methods to make structured structures out of this. Yeah, but it can be quite advantageous. On the other side, you are taking a polymer, you're reinforcing it, right, with a cellulose nanocrystal. So extrusion is more difficult, for example. So yeah, things like laser sintering can be quite interesting, or laser cutting.
So will the 3D printing, the rod will fit the needle?
Yes. So for the cellulose nanocrystals, we're talking about, the very longest ones are, say, 4. So that has no problem clogging, or it won't clog the needle if done in the right way. And most of the commercially available cellulose nanocrystals are about 250 nanometers long. So yes, they change the viscosity, but no, they won't get stuck in the needle tip.
Okay, thank you very much.
All right. Thank you. Next panelist, Zhuo Li.
Hi, Johan. Very good talk. Actually, I also have some questions regarding the mechanically adaptive composite materials. I think it's interesting to see the sea cucumber example that it's adaptive to the environment. So what is the factor that triggers your CNC PVD composite to change the modulus? I saw you show orders of magnitude change in modulus, but what is the mechanism for that?
So in that case, in the dry state, so outside of the body, the cellulose nanocrystals are held together with hydrogen bonding. So this is inside of a polymer. To the previous question, the polymer has to swell a bit, or has to be able to swell a bit. When we implant it inside of the body, so you push it into the brain, that composite takes up a little bit of water, decouples the interactions between the cellulose nanocrystals, and that's what causes the order of magnitude decrease in the mechanical properties. So it's really swelling, so water transport into the composite, and then decoupling of the cellulose nanocrystals, decoupling of the hydrogen bonds between the cellulose nanocrystals.
Does that require a lot of water, or have you quantified the amount of water that's needed?
No, we've never actually quantified the amount. For other applications, like we've looked at trying to make car tires that switch their mechanical properties when they get wet. We have looked at how much time it takes for the water to get out of the system, because there's surface water, and then there's water within the general material, and then there's water bound to the surface of the cellulose nanocrystals. So we have looked at the amount of time, but we've never actually looked at quantifying the amount of water that's required inside of that.
Yeah, just because I'm curious, put it into the brain. So maybe I'm not sure how much water the brain can supply to those probes.
Think that the electrode is pretty small relative to the overall the amount of brain tissue, and so it wouldn't denature, or wouldn't dehydrate the surrounding tissue. And it really happens over the course of, say, 5 to 10 minutes. So that would allow water transport from the rest of the body for sure.
I see. So is the drag release mechanism more or less related to the hydrogen bonding between the CNC as well?
Yes, the mechanism that I showed in this talk was simply just kind of adhering the drugs to the surface of the cellulose nanocrystals, and then allowing diffusion to take them out of the composite. We have used like a covalent bonding, like say a Michael addition and then a retro Michael addition to remove it. So that would be acid or base catalyzed. But in this case, no, it was just simply adhesion to the surface.
And another question actually come up to me when G asked the first question about the 3D printing thing. So because of the shape of the CNC is in the shape of like a rod. So have you ever think about making it into some viscosity modifier, or does it have any kind of liquid crystalline like properties, because they really look like those rod-like shape?
Yeah. I mean, my PhD was in liquid crystals, but thermotropic liquid crystals. Cellulose nanocrystals. So I show them as perfect rods. They're not. They're actually a twisted rod, and so in water, for example, they do form a chiral pneumatic phase if you have them at the right concentration. And our group doesn't usually utilize this, but you can take that chiral pneumatic structure, template other structures onto it. So, for example, silica, and make some very nice colored structures because of that chiral pneumatic structure. So it certainly is possible. And yes, they do act as viscosity modifiers. We've used them as viscosity modifiers in like drilling muds. But there's a lot of people that use them in, say, cosmetics and other applications like that, in kind of the very like colloid based systems.
Yeah, I think that's all for my questions. Thank you. It's nice to see Hokies again.
Yeah, go Hokies.
Go Hokies.
Very good. So we can move on to Professor Zhang.
Hey, Johan. This is Xiaosheng from Chengdu. So thank you so much for the wonderful talk. It's very, very interesting. In my lab, we also have some work focusing on the cellulose materials, but most of this work is about electronic devices. So my question is, can you give some comments on the future of introducing this cellulose nanocrystals into the cell destroying devices, especially the transient electronics? So since cellulose is also well known as a promising biodegradable materials. Of course, we already talked about the biodegradable materials, but I'm wondering, for this the biodegradable features. So is it, how about the controllability and which is also very useful to construct this the transient devices. So can you give comments and what is the advantages and disadvantages if we want to use these cellulose materials to form this kind of the self-destroying devices? We can control the lifetime of this kind of device, especially for the electronic devices. Thank you.
Yeah, I think there's a lot of potential application in electronics. I mean, in one respect, cellulose is a horrible material for electronics because it's non-conductive. But that is why it's also being used, right? I mean, printable electronics onto paper, for example, great. Again, modification of the surface. You can do some very neat things. The biodegradability of cellulose is fantastic. Not within the human body so much, I mean, because we can't digest cellulose, but in nature, there's a lot of bacteria and a lot of processes that will degrade cellulose. In its simplest form, it is a very simple polymer backbone, and I think will always be inherently very biodegradable. You need to really modify the surface of these materials, say with a lot of silanes or a lot of like hydrophobicity, before it will become not as biodegradable. So I think the degradability in nature will always be there. That being said, I think to the electronics, you want to be able to degrade this within like and extract out the other components of the electronics. And so the thermal stability we've talked about, the stability in acid would probably be the method that I would use. You know, using a strong acid or a strong base would quite simply dissolve the polymer, which would allow you to access, say, metals or for other components of the electronics. So I think, yes, I think it's an inherently very degradable system, which for short-term electronics would be very advantageous. For long-term electronics, I don't think cellulose is what you'd want to use. Again, because of that degradability, right? Because of the swelling of the material, for example. If it's not modified, it will uptake water, and then that degradability will happen even quicker. So I think on one side, for short-term electronics, very good. I think the degradability is well proven by millions of years of of evolution. But on the other side, yeah, I think you can control the degradation using very simple chemicals. So you could extract out anything you wanted.
Okay, thank you. So I have a very short question about the manipulation of this cellulose nanocrystals. So is it possible to precisely manipulate the CN and to form the design and microstructures or surface? I mean, in smaller scale, I know that you already developed the 3D printing technology to manipulate these materials to form some micro scale structures. But I mean that at lower scale, like maybe we can. Is it possible to manipulate these the cellulose nanocrystals, for example, like the self-assembling, and we can assemble this nanocrystal one by one, and maybe you need to surface modifying the surface and by some functional the groups, and then to cross-link them to form. Is it possible or not? Have you tried? Okay.
I think on an individual cellulose nanocrystal level, that's very difficult, right? I mean, manipulating a single cellulose nanocrystal would be quite difficult. That being said, we do have a lot of tools to be able to talk about self-assembly, right? The functionalization of this, I mean, I kind of oversimplified the structure of this. The ends of the cellulose nanocrystals are you can functionalize specifically the ends of the cellulose nanocrystals and not the rest of the surface. So that would be one tool. We talked about the chiral nematic nature of the cellulose nanocrystals, so you can get them to self-assemble into helical structures. And then on the other side, you can put them in an electric or magnetic field and get them to align. So there's a bunch of tools that we can use to manipulate them. I would hesitate to say on a single cellulose nanocrystal level, but on a self-assembly level, then yes, I think we could. So it would be it's very interesting to potentially make very, very small structures from these.
Okay, thank you so much.
I should follow up to that question, Johan. Do you think the electrodes you're making are also sensors? Are they pressure sensors? Because if your cellulose is swelling, right?
Yeah,
it would stretch the surface area that you have, like the contact.
Not in the traditional sense of, I mean, you wouldn't get an electronic output of from that. However, I mean, you would get an output. If you squish these materials, water would come out, and you should be able to change. If you were to take a conductive cellulose nanocrystal inside of a composite, and you compressed it, you would get a change in the electronic properties. So that would be detectable. So yes, you could create, and like if you made a chiral nematic structure, you would compress the chiral. That helix, and you would get a change in color or a change in. So yes, you could make some sort of sensor out of it.
Because I'm thinking, especially for the brain electrode that you're using, if you're using a very thin film of gold, it doesn't take a lot of stress to expand the surface area, and that surface area is proportional to the amount of current you can get. Right. So if I stretch my film, even if it's by 1%. At that level, I should see a significant change in conductivity. So maybe you're making some new types of MEMS, not based on piezoelectric behavior, but actually based on the shear coupling between your film material and whatever conductive system you have. And if you have not explored that might be a very interesting way of talking about new types of MEMS that could actually even detect osmotic pressure other than mechanical pressure, which is important in systems like brain and close tissue.
I don't know if I would choose polyvinyl alcohol to do that, just because it's not very stretchable, but we could do something like polyurethane, right? That would inherently much more stretchable. Obviously not as water sensitive. So you'd have to balance that. How much does it swell versus how much can it stretch? To make that an effective sensor.
Talk to Professor E in the panel. She does artificial skin, and I think there's an opportunity there for a collaboration where you can both look at the stretchability of her systems under the influence of your reinforced materials, and maybe you might be able to find a new type of MEM sensor. Who knows?
For sure. Yeah, that sounds very interesting.
Because I have some of our papers in this field. Actually, it seems like they could be very well coupled with what you're doing.
Yeah, no, we should definitely talk and see what we can do there. It's not something that I've thought about doing.
Yep, perfect. And then you can continue the Hokie partnerships.
Yes, exactly. Yes, Hokies around the world.
Yep.
For everybody that doesn't know, a Hokie is the mascot of Virginia Tech, and so that's why we're talking about Hokies. Very
good. All right. For the interest of time, I think we're going to move on. And thank you so much, Professor Johan, for that very wide breadth of conversation on a material that we all know, but we quite don't know, cellulose. And was this in a normal like a in-person presentation, I would walk across the stage with Alice and would hand you the certificate, but now please accept our virtual. It's going to be emailed to you. You've graced our 110th volume of the ICANX. And to everybody, I want to make a few announcements. One is next week, sorry, this is for this week. Next week, we'll continue the conversations on our bio-inspired material. And I will invite all of you to come and join us. We also have ICANX Young Investigator Awards coming in. I was not able to put a slide in time, but please encourage all the young investigators to join in. Thank you very much. And have a good one, everyone.
不再是奇迹，不再是幻想，此刻纯感觉全世界对我鼓掌，不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱，我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞，不再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中